Hi, welcome to the Sybil Protein Report Page screencast. This screencast will cover the features of the Sybil Protein Report page. The Protein Report page can be accessed from several places in the Sybil system. For this demo, let's do a protein search. We'll start at the Strep Pneumo homepage at strepneumo-sybil.igs.umaryland.edu. From here, I'll click on the Protein Cluster Search button. Let's search for proteins with the word protease in the name, and for proteins that have a transmembrane region. Now I see my results here. Let's click on SP3059060631, a serine protease. This will open up a protein report page in a new tab. Depending on your browser, this may open a new window, but in Firefox, it should open a new tab. The Protein Properties table has basic information about the protein, including the source organism, gene product name, polypeptide length, and the date the annotation was created. The Database References table contains alternate accessions for the gene. The Protein Clusters table lists the clusters that this gene is a member of and provides a link to the cluster report page. The Genomic Context table gives the name of the genomic sequence on which this gene resides, along with its location and strand. The image that appears gives a visual representation of the gene and the surrounding area. Additional feature types can be added to the image by checking the boxes in the Additional Feature Types area. I'll click Select All for this image. The amount of padding on either side of the gene can be changed by clicking one of the available pad values. Let's try 10 kb on either side of this gene. Genes are depicted as rectangles with arrowheads indicating their strandedness. Very short genes have an actual arrowhead protruding from the gene rectangle. Features in the picture can be clicked on for more information. For instance, we can click on one of the neighboring genes to find out more about it. If I click on the neighboring gene SP3059060634, I'll get a pop-up. This pop-up has the name, which doubles as a link to the gene's protein report page, gene product, links to the comparative view, which will be covered in another section, download buttons for the nucleotide and protein sequences, and a list of clusters that this gene is a member of. The cluster names double as links to the cluster report page. Below that is a list of sub-features. These are predicted features of the gene that have been drawn as glyphs on top or just above the gene. Each feature has a location, a brief symbol description, and sometimes a longer description detailing where the feature came from. In this case, the gene has a predicted signal peptide, a cleavage site, and several transmembrane regions. The pop-up window can be moved around by clicking and dragging the title bar. And then the window can be closed by clicking the X on the top right. Features other than genes can also be clicked on. For instance, clicking on one of the blue rectangles will bring up a small pop-up telling us that we clicked on a box C repeat and what the repeat's precise coordinates are. The BLAST P hits area contains a graphical representation of the best BLAST hits for this protein. The query protein is shown at the top of the graphic, and the hits are shown below. Each hit's high-scoring pairs are drawn. The name of the hit gene is shown above the HSP graphics, and the E-value and percent identity for each HSP are shown just below the HSP drawing. Clicking on a hit will give you a pop-up just like the ones in the context image above. The only difference is that the Fmin and Fmax represent the coordinates of the hit in the protein sequence instead of the location of the gene in the genome. More blast hits can be shown by clicking a value next to the words Show Top. I'll select 50 to show the top 50 blast hits. Also, I'll select the cluster name next to the words Highlighted Proteins to highlight all the hits that are clustered with the query protein. As we scroll down, we can see that there are a number of high-quality hits that ended up clustering with SP3059060631. The 22nd hit, however, appears to have been a high-quality hit but only covered approximately half of the query protein. This protein did not cluster with our query. As we scroll down further, beyond hit 33, we can see that the quality of the hits drops dramatically. We also see that there are several small HSPs that aligned but did not produce a full-length match. The last section provides the protein and nucleotide sequences of the gene. The sequences can be downloaded using the buttons, or copied and pasted right from the browser.